It's a great pleasure to be joined by Dr. Kate Duchowny, who is a social epidemiologist and a K99 funded postdoctoral scholar in the Department of Epidemiology and Biostat at UCSF. Kate's overarching research goal seeks to bridge the social, environmental, and biologic determinants, interestingly, of musculoskeletal health and physical functioning in older adults, with a particular focus on informing interventions, which is quite interesting. Look forward to hearing more about that. Her current research is organized around two lines of inquiry. The first is identifying which aspects of the built and social environment matter most in helping older adults maintain independence and physical mobility. And the second line of inquiry is examining life course sociobiologic mechanisms, things like viral infections, mitochondrial function, et cetera, that drive disparate outcomes in physical disability, especially related to neighborhoods, which is something that is near and dear to my heart as well. So without further introduction, I want to turn this over to Dr. Duchowny.
Thank you, Dr. Brannis, for that very warm introduction. I'm going to go ahead and share my screen. Can you all see my slides okay and hear me okay?
Yes, they look great.
Great. I'm just going to configure my Zoom screen so I can have a few familiar faces and warm faces to see while I'm talking into the computer. So again, thank you so much for the warm introduction and for the opportunity to come and speak with you all today. I'm so bummed I can't be there in person, but I'd like to thank Dr. Brannis for the opportunity to come and speak, as well as to Gerald Govia for facilitating my talk that focuses on uncovering the intersection between social determinants and musculoskeletal health to promote health equity. And I'd like to just ask the Zoom audience to hold their questions to the end. So I'd like to just give sort of an overview as to where we'll be heading today. I'll first be providing a little bit of background about me, and then a sort of broad overview about aging research and making the case for why musculoskeletal health is so important. I'll then be highlighting some current work from 3 different projects, and then I'll be concluding my talk by discussing future directions as well as my planned research. So I'd like to begin by telling you a little bit about me, who I am and where I've been, and how the journey that I've sort of been on to date has really shaped the work that I do today. My work sort of begins at the University of Miami, where I first received my bachelor's and master's in public health. And it was during this time as an MPH student that I developed a passion for community-based public health research. And that's what actually brought me to New York City, which I view as the true jumping off point for my career in public health. I joined the New York City Department of Health as an independent restaurant coordinator, which was part of a CDC-funded initiative that looked at innovative ways to reduce sodium in the independent restaurant environment as a way of preventing CVD. And it was really through this work where I basically, over the course of 3 years, I was on every single subway line from one end to the other across all 5 boroughs. And it was through this work that it brought me to various locations and neighborhoods all throughout New York City. And it was through this that I really gained sort of a deep appreciation for the inextricable link between health and place. And that is really what motivated me to return to school to pursue my PhD in epidemiology at the University of Michigan School of Public Health, where I joined the Center for Social Epidemiology and Population Health. And it was during this time that I gained an even deeper understanding of just how important neighborhoods were for health and well-being, and specifically the role of the built neighborhood environment. As part of my work as a PhD student, I examined to what extent the neighborhood built environment, specifically features like uneven sidewalks shown here, curb cuts, potholes, and other aspects of the built neighborhood environment influence mobility. And it was during this work that I really started to understand just how important neighborhoods are for older adults to age in place, and how neighborhoods directly influence mobility disability in older age. And so I shifted my sights to aging research, which allowed me to blend two areas of passion of mine, social epidemiology and older adult health. I brought these two areas of interest with me to the University of California, San Francisco, where I am now in the Department of Epidemiology and Biostatistics. First as a T32 postdoctoral trainee, and now as a K99 postdoctoral scholar. I'd like to begin by discussing my conceptual framework that really motivates the work that I do. And I'm going to spend a little bit of time here sort of walking through this framework in detail, both holistically as well as discussing these individual domains and their relationships, and highlight where I see major gaps existing and how I see my larger work situated within this conceptual figure. And so ultimately, by the end of today's talk, you'll be able to see how I'm working towards really bringing these domains together. But first, before I dive in here, I want to just focus right here on health inequities, sort of where we'll begin. And that is, I want to call attention to the fact that I use the terms health inequities and race throughout my presentation. I want to sort of have an aside here with you all about what I mean when I use those terms. First, with regard to race, really foundational work by giants in the field like Dr. Kamara Jones, Dr. Rhea Boyd, Dr. Chandra Ford, Dr. Zinzi Bailey, and others have really paved the way in helping us think about within the field of health equity, what we mean when we talk about race. Race is a socially constructed variable that reflects a complex process that is both rooted in and determined by structural racism. So when I say, for example, that I enter race into a statistical model, what I mean by that is that we're using this variable as a way of capturing variation in the lived experience, which we know is highly differentiated across the life course. I also use the term health inequities throughout my presentation. I use this term in sort of different to the idea of a disparity, which isn't just merely a difference. I see health inequities as those differences that are not only unnecessary, but also avoidable, and therefore are considered to be unjust. So as long as medical records have existed, so too have health inequities. Health inequities have persisted for centuries, and yet we've been unable to meaningfully intervene on these inequities in order to move towards health equity. Part of the reason we've been unable to achieve this goal is that there's been a singular focus on biomedical risk factors. And certainly, while we've made tremendous progress in really understanding how biomedical risk factors shape health status and influence health inequities, we have not closed the gap, as evidenced by the fact today that gross inequities still continue to persist. At the same time, on the other side of this pathway, moving more upstream, we've made huge gains with regard to our understanding of the social determinants of health. And of course, there's a vast body of literature that has repeatedly shown that the social determinants of health influence downstream health and shape health inequities. For example, we know that there are far-reaching effects of structural racism that shapes everything from neighborhood resources, access to education, occupational opportunities, to name a few, and that these experiences are highly differentiated across the life course and serve to exacerbate health inequities. However, one difficulty in doing this work is that we have yet to identify the underlying biologic mechanisms that would help us understand how social experiences get under the skin and contribute to inequities, as depicted here by the black box. I'd now like to focus in the middle part of this pathway and talk about muscle health. I've chosen to focus on muscle health because it is such a strong marker of current and future health status and is related to numerous aging-related outcomes. And it is a highly determinant of the ability to age independently in older age. And yet, despite the outsized role that muscle health plays for health status in older age, we find ourselves in a similar place still, where the majority of work that's been done to date has been conducted within a biomedical risk factor framework. And again, here, while we've certainly made huge progress in understanding, for example, how obesity influences muscle health, we have failed to document and intervene on health inequities and intervene on musculoskeletal health inequities. Part of the reason for this is that we have this black box on the other side of this pathway, where we have yet to fully acknowledge and integrate the social determinants of health into the study of muscle health as a way of closing the gap in health inequities. So this really begs the larger question, where does this leave us? This is where I view my contribution and how I see my overarching conceptual framework. I see my work as an attempt to unify these domains as a way of understanding, disrupting, and intervening on health inequities in order to improve adult health. I set my work within a life course framework as depicted here by the arrow on the bottom, because really what this work requires us is to examine and measure salient exposures throughout the life course from childhood all the way until adulthood. And prior research shows us that we actually cannot simply afford to pay attention only to the right side of this pathway, because we already know what this produces: health inequities that still exist today, and we will be unable to meaningfully intervene until we are able to bridge this gap of really bringing all of these pieces together. I'd like to point out here that there are 4 large separate arrows here, and of course, many other arrows that reflect complex multifactorial processes that I'm not showing here for the sake of simplicity. But what this means is that in order to do this work, some of the research I will be presenting today will take up only one of these arrows, or will focus more upstream or downstream than others. But the larger project stays the same for what I do. And that is, all of my work can be conceptualized as fitting into this larger process that links social determinants to health equity. And so throughout this presentation, I'll be continuously referring to this conceptual framework as a way of orienting you all as to which part of the conceptual figure I am referring to. So today I'll be presenting work from 3 projects and an example from 1 paper from each. I'll be beginning with my first project, which seeks to define cut points for clinical muscle weakness. I'll then be presenting work that examines the relationship between hand grip strength and brain health. We'll be moving more upstream to examine the relationship between cytomegalovirus and physical disability. And I'll conclude my talk by discussing my current K99 R00 award, as well as discussing my planned research. But first, I'm going to segue and talk about older adult health and why musculoskeletal health is so important. We are currently undergoing a massive demographic change where the older adult population is rapidly increasing. And the reason why this is so important is that it coincides with an alarming demographic trend. And that is that mobility disability is actually increasing among middle-aged adults. And the reason why this is so important is that in just a mere 9 years, all baby boomers will be age 65 years and older. So what this means is that we're undergoing this massive demographic shift, both in terms of the aging population, but we may also be on the precipice of observing a huge increase in mobility disability, which does not bode well for the maintenance of physical functioning and independence in older age. One very important determinant of mobility disability is muscle weakness. To zoom out a bit, muscle weakness is a normative part of the aging process. And in fact, I'm sorry to say, it usually begins around 30 to 40 years of age. And we're all undergoing some aspect of decline, but we know that around 50 to 60 years, there's an acceleration that typically occurs. Luckily, one really great thing that we have going in our favor in really measuring total body muscle weakness is that hand grip strength has been shown to be a surrogate, reliable measure of total body muscle strength. So what that means is that if you know an individual's hand grip strength, you have a pretty good picture as to what their total body muscle strength is. And in fact, there's research to support hand grip strength as a robust predictor of future physical functioning. In fact, in one study that looked at hand grip strength at baseline among 4,000 men, those men that fell in the lowest tertile had reduced physical functioning 25 years later. And so really hand grip strength is this robust predictor and also may be reflective of some subclinical process that has yet to fully manifest. And then it's picking up on this earlier in the life course. Muscle weakness is important for many health conditions. It's been shown to be associated with mortality, dementia, stroke, cancer, cardiovascular disease, physical disability, and frailty. And I want to touch on this point about frailty. And that is because, of course, Dr. Linda Fried has really pioneered the Freed frailty criteria, really thinking about how we understand frailty, how we measure it. And I'm showing here these the 5 criteria of this phenotype. And what I'd like to draw your attention to is that weakness is listed as a criteria for the measurement of frailty as measured by hand grip strength. And there's been work that has repeatedly shown that, in fact, muscle weakness on its own is a robust marker of frailty, even in comparison to the other indicators of frailty. And so it really is this powerful marker of frailty in physical functioning. Here I'm showing normal grip strength trajectory. This is trajectories for across the life course. So this is data from 12 different population-based studies in England, making up close to 50,000 people, men on the left and women on the right. And as you can see, while there's certainly a normative trajectory for grip strength, there's also a considerable amount of variation. And understanding what may be driving this variation, excuse me, is important. And we've actually done some work to examine this. So given that we know that neighborhoods are so important for physical health, we reason that neighborhoods with greater physical disorders, so these are neighborhoods where participants report having there's more litter on the ground, there's vandalism, vacant lots, those neighborhoods with higher levels of physical disorder would be associated with lower hand grip strength. And that is exactly what we found using data from the health and retirement study. Okay, so now I'd like to shift to my first project, which was born out of my dissertation and sought to address a very basic question, which is, how do we define muscle weakness? So despite the fact that we know hand grip strength is consequential for older adult health and functioning, there have actually been major disagreements in the literature regarding how best to define clinical muscle weakness. And there was no universal agreed upon standard. And so in 2010, as a way of trying to gain consensus regarding how best to define clinical muscle weakness, the Foundation for the National Institutes of Health, also known as the FNAH, took on this project as a way of proposing a universal gold standard. And the way in which they went about doing this is that they pooled data from 8 different RCTs and 6 cohort studies to define muscle weakness via hand grip strength cut points, shown here on the left for men and women. A major limitation, however, of the FNH pooled data source is that it was 98% white. And yet, the cut points that they derived are considered to be the gold standard in clinical practice for everyone. Therefore, we were really interested in establishing cut points for muscle weakness in a diverse sample of older adults, and then subsequently to take these cut points and to quantify the prevalence using nationally representative data, and then to compare how our cut points would fare to that of the FNAH. In order to do this study, we used data from the health and retirement study, and just briefly here about HRS. HRS was established in 1992, and it is the longest running longitudinal study of American adults age 50 years and older. And participants are followed every 2 years. And when weighted, it's considered nationally representative. Beginning in 2006, there was an enhanced face-to-face battery that was instituted within the HRS where they obtained physical measurements. And for the purpose of this analysis, this is where we obtain our measurements for hand grip strength and walking speed. We used data from 2010 and 2012, and we restricted our sample to those aged 65 years and older and essentially replicated the exact same methods that the FNAH used in order to make comparisons. Our key variables of interest were the maximum value of hand grip strength as a proxy for total body muscle strength. And then our calibrating variable, which I'll explain further, was mobility impairment. You can think of this as our primary outcome, which is having a slow walking speed of less than 0.8 meters per second. So in order to do this analysis, we use classification and regression trees. Just briefly here about CART. CART is a non-parametric decision tree learning technique. And the way in which CART works is that it creates decisions based on what variables are most closely related to the outcome. They're commonly used in medical decision-making. However, more recently, they've been used in DNA sequence analysis, genetics, and epidemiology. CART in general is considered a relatively new tool within the field of social epidemiology. So the variables that were entered into our model were our calibrating variable, or our primary outcome was slow walking speed. And then we entered hand grip strength, age, chronic conditions, BMI, and self-reported race. Before I get into the results of this finding, I just sort of walk through how CART works, a little sort of toy example here to show you how trees are derived. So in practice, the way in which CART works is that a data set is recursively partitioned until it finds a cut point that best discriminates subpopulations based on the outcome. So what does this look like? So here we have a 2D data set on the left, and CART finds the cut point that best discriminates here. So we partition our data space, and then it's recursively partitioned until CART detects that there are no further gains that can be made, or there's some preset stopping rule that's met. And so here I'm showing you, this is just one sort of purity index. This is a criteria that's used to maximize the information gained at each split. This is the Gini index. And essentially what it does is that I use the Gini index as a way of optimizing the balance between the number of nodes that I have and then the number of observations within each node. And so you can imagine, you know, in theory, I could have recursively partitioned this data set until I hit every single data point. But of course, that's not really helpful when we think about prediction. And so we need to balance the number of nodes with the number of observations. And one way that we do this is in the form of pruning. And so pruning is implemented. You can imagine, of course, this is a very simple tree, but you can have these very, very large complex trees. And so we prune our trees as a way of removing subnodes and to reduce the complexity of these trees to make them interpretable, and also to improve predictive accuracy and to avoid overfitting. And so now returning to this project, these are the variables that we entered into our model. And I want to make a note here that we enter in race. Again, we use this variable as a proxy of really capturing the racialized experience across the life course. And interestingly, we had primary splits on race, which indicated to us that we should be running subgroup specific trees. And so that's how we proceed. So I'm showing you here the CART results for by subgroup, and a few notes here before we walk you through one of these trees. We had primary splits on hand grip strength, which was sort of in line with what we expected, given the relationship between muscle weakness and slow walking speed. It really does discriminate in terms of understanding who's weak relative to who's slow. And so that's the primary split. We did have subsequent splits, but I'm not showing them here just to keep things streamlined. And so I'll let you all sort of look at these trees, and then we'll walk through an example. So here, I'm just going to zoom in here on the tree for Black women on the top left. So in our sample, we had 635 Black women, and the prevalence of slowness was 88%. CART determined that the hand grip strength for Black women that best discriminated was 31 kilograms, shown right below. So these are the hand grip strength cut points. And what you can see is to the right of the right side, those are the women who fell below 31 kilograms. So 88% is quite high, and I'll be returning to this point. So after we derived our cut points, we were interested in understanding the prevalence of muscle weakness in HRS. And so what you can see here overall is that the prevalence of muscle weakness is quite high across all groups, but in particular, we notice a disproportionate burden among Black women and Black men. We were then interested in understanding to what extent our cut points would be similar or different to that of the FNH. And so what you can see here shown on the left in green are the cut points, are the FNH cut points in quantifying the prevalence of weakness in HRS. And then on the right in blue, these are our cut points, the HRS cut points, quantifying the prevalence of muscle weakness in HRS. And so most notably, what these numbers really show us is that if we look on the left, the prevalence of muscle weakness is 9 to 13% using the FNH cut points, and 47 to 55% in HRS. And so really what this tells us is that the FNH cut points misses a whole group of people who would be considered weak using the HRS cut points, but are otherwise going undetected. And of course, this has major implications in thinking about who we're classifying as weak and who we're going to be targeting for interventions as a way of reducing disparities. This is a really important and stark finding that we observed. To conclude, we derived cut points for clinical muscle weakness using nationally representative data. And we found that our cut points were very different to that of the FNAH, which are currently considered the gold standard. I've done some work to look at how predictive our cut points are of future health status. And in a paper that I published in 2019, I found that individuals who were considered weak at baseline as defined by our HRS cut points, they had a decreased survival across a 9-year period compared to those who were not weak. Lastly, I want to return to perhaps the most important point and finding of the study is that we observed a high prevalence of muscle weakness across all groups, but in particular, a stark inequities with regard to muscle weakness among Black men and Black women. These findings were surprising and really warranted sort of a deeper dive and really understanding how social experiences may be embedded as a way of driving these disparate outcomes in later life. And so as a way of trying to answer this question, I led a study that was published in the Social Science and Medicine Population Health that sought to answer a question as to what extent life course trauma may influence grip strength trajectories in later life. And what we found is that individuals who underwent greater life course trauma earlier in the life course had steeper declines in their hand grip trajectories in later life. So I'll now be turning to my second project, which sought to examine the relationship between hand grip strength, dementia risk, cognition, and neuroimaging outcomes using data from the UK Biobank. So briefly here, one of the hallmarks of dementia, Alzheimer's disease, and other pathologies is a decline in cognition, specifically in memory, and a decline in functional abilities. We know that physical functioning may be an early indicator of brain changes. There's literature to suggest that lower hand grip strength is associated with poorer cognitive performance. And this is, this makes sense given that we know that physical functioning is tightly connected to brain circuitry and that they have shared pathology. However, despite the work that's really been done in this area, there's really been sort of some key methodologic challenges in really making sense of the results to date. The first is that there's been a lot of inconsistency with regard to study design. There's been a lot of cross-sectional work, and of the longitudinal work that's been done, a lot of it is varied in follow-up time and duration. And a lot of these studies have been done in sort of small, select clinical samples that really make generalizability difficult. Additionally, a lot of the work that's been done has focused on maybe one domain of brain health, so one neuroimaging indicator. And there's also been an over-reliance on cognitive screens like the MMSE, which are not really specific in terms of understanding sort of the underlying mechanisms about what could be happening. And so for our study, we sought to examine whether hand grip strength is associated with reliable markers of cognition, neuroimaging, and incident dementia in a large prospective population-based cohort. We use data from the UK Biobank, which is comprised of over 500,000 adults aged 39 to 73. And for our analysis, we only included those who were dementia-free at baseline. Of note here I just want to call out about the UK Biobank is that one of the limitations is that it is predominantly white. And although we did look at racial differences, we did not find any. And so what I'll be showing you here today are racially pooled results that are stratified by gender, given known differences in hand grip strength. Our exposure of interest was hand grip strength as a proxy for total body muscle strength. And our outcome was actually comprised of sort of 3 domains of outcomes, which I'll walk you through now. So our first group of outcomes were with regard to incident dementia. We looked at all-cause dementia, vascular dementia, and Alzheimer's disease. The second group of outcomes was total brain volume, hippocampal volume, and white matter hypertensities, and these made up our neuroimaging indicators. And for the last outcome, we looked at 2 cognitive measures, fluid intelligence and prospective memory. And really what these outcomes offer together is that they're sort of complementary with respect to prospective measurement and also offer sort of unique insights with regard to sort of biologic mechanisms. So in order to analyze each of these group of outcomes, we had to employ various statistical approaches. So for the first group of outcomes, we employed Cox proportional hazard models where we treated death as a competing event from the death registry. And then for our second group of outcomes, we used mixed effects linear regression where we entered a fixed slope and a random intercept to account for repeated measures within individuals. And for the last group of outcomes, this was a cross-sectional analysis, but we employed mixed effects, linear and logistic regression. And so as you can imagine, there's a lot of outcomes here. There were not a lot of analyses that we ran. So I'm just going to be providing sort of a high level overview of what we found in the interest of time. So first, we find that hand grip strength is associated with incident dementia. And I'd just like to draw your attention to the results on the far left. And specifically what we found is that for every 5 kilogram decrease in hand grip strength, it was associated with a 17 to 20% increase in the hazard of all-cause dementia among men and women. Second, we found that hand grip strength is associated with fluid intelligence and perspective memory. So on the left here, we see that for every 5 kilogram decrease, there was a reduced odds. On the left, we see that for every 5 kilogram increase for the fluid intelligence models, we see that there's a decrease in fluid intelligence, both for men and women. And on the right, we see that again, for every 5 kilogram decrease in hand grip strength, there was a reduced odds in answering the perspective memory question correctly for both men and women. Lastly, we found that hand grip strength is associated with neuroimaging correlates. Specifically, we found that a 5 kilogram reduction in hand grip strength was associated with lower total brain volume, reduced hippocampal volume, and an increase in white matter hyperintensities. So to conclude, we found that hand grip strength is highly associated with incident dementia, neuroimaging correlates, and cognition in a large longitudinal cohort in the UK. So some future directions of this work is that we would be especially interested in obtaining data, PET data, as a way of sort of drilling down on mechanisms with regard to the Alzheimer's disease findings, as a way of investigating the role amyloid or tau may play in these findings. And then we'd also be interested in replicating the cognitive models longitudinally. We unfortunately were only able to complete this analysis using, it was a cross-sectional analysis. So for my last study, we'll be moving slightly more upstream to examine the association between cytomegalovirus and physical disability by race, ethnicity, and gender in the health and retirement study. And this work is currently in press in the American Journal of Epidemiology. So before we dive in here, I'd like to begin by sort of asking a basic question, which is, why should we study cytomegalovirus? Well, first, CMV is actually believed to be a lifelong infection, and it's estimated that about 50 to 80% of adults have been infected by age 40. And so what this means is that once you're infected, the virus is in your immune system for life. And so we know that on average, most of the infections that do occur happen in childhood. Important for the work that I do, we know that CMV exposure is heavily socially patterned. And what I mean by this is that recent research suggests that individuals who occupy greater social disadvantage are believed to have more exposure to and greater reactivation to the virus. And so because CMV is a lifelong infection, there are these cycles of latency and reactivation that can occur. And so I'd like to just continue on with this point by showing this figure here to sort of explicate what this looks like. So you can think about a primary infection, again, most often happening in childhood, and that there are these cycles that can occur throughout the life course whereby there's reactivation and then replication of the virus. And then at each of these points, the immune system has to dedicate all of its resources in getting the virus back under control, and that this process can repeat itself. Recent research suggests that one potential factor that influences cycles of reactivation is life course stress. That is, the more life course stress an individual experiences, the greater the number of reactivation cycles that can take place. And within each of these reactivation periods, again, the immune system has to dedicate major resources to really tamping down the virus. So why is this important? Well, what this means is that we can think of CMV as both a social and biologic exposure. And that when we think about how to measure this is that CMV IgG antibody levels may serve as sort of an imperfect proxy of immune control and an overall indicator of immune fitness. So in other words, we can think of CMV antibody levels as sort of providing a window into how well the immune system is doing in keeping the virus under control. And the reason why this is so important is that CMV antibody levels may therefore serve as sort of a potential model and understanding how social stress imprints on the immune system. And ultimately, how this constant wear and tear on the immune system through these repeated cycles of reactivation may lead to accelerated biologic aging. So, given longstanding inequities in physical disability, we sought to examine to what extent CMV seropositivity is associated with greater prevalence of physical disability and whether or not this association would differ by gender and race ethnicity. Our hypothesis was that CMV seropositivity would be associated with a greater prevalence of physical disability in comparison with seronegative individuals, and that perhaps this relationship would be stronger for racial and ethnic groups and women due to greater social disadvantage experienced across the life course. In order to answer this question, we used data from the health and retirement study, specifically the venous blood sample from 2016, which was composed of 9,000 adults aged 50 years and older. Our exposure of interest was cytomegalovirus, where we used cytomegalovirus IgG antibody levels as measured in blood serum, where we compared those who were seropositive to CMV to those who were seronegative. Our outcome of interest was activities of daily living disability, and where we compared those who reported difficulty in one or more activities of daily living, such as dressing, bathing, toileting, and then compared those who reported one or more difficulty to those who reported none. We employed Poisson log binomial models with robust standard errors in order to obtain a prevalence ratio, and then we entered interaction terms for gender and race ethnicity. Of note, we did not find any statistically significant interactions for race and ethnicity, but we did for gender. And so I'll be presenting gender stratified results. So first here, I'm showing you the results for men. What you can see here, there were sort of a bunch of models, but I'm just showing here the age adjusted sort of crude model and then the fully adjusted model. We adjusted for BMI, chronic conditions, and a host of other covariates. And what you can see here for men is that it's pretty much a null story. In contrast, again, after adjusting for age, education, smoking, health conditions, BMI, in this fully adjusted model, is that what you can see is that our results suggest that overall, women who are seropositive to CMV had a 14% higher prevalence of disability shown here. So to conclude, we found that CMV is associated with a greater prevalence of disability among women, and that perhaps CMV may serve as a salient mechanism in really shaping disability risk in older age or middle age and older adults. One of the things that gets me most excited about this paper is that it provides insights into one day how we may be able to potentially intervene on physical disability by targeting the immune system for interventions, either in the form of a CMV vaccine or some other immunotherapy that target immune aging pathways. So I just want to wrap up here about what I've shown you today. I showed you a paper that derived cut points for clinical muscle weakness. I made the case that hand grip strength may be consequential for brain health. And then we moved upstream where I showed you some evidence to suggest that CMV may be an important risk factor for disability, in particular for women. And so I now like to talk about the last part of my talk here about my current and future directions. So there's general consensus that we've sort of advanced our understanding in each of these areas of study over the last decade. And I've shown you today that how I'm working in sort of each of these domains to chip away at these larger bodies of work. However, what's needed in order to really bring these domains together is an interdisciplinary approach in which these areas become truly integrated so that the sum is bigger than the parts. And ultimately, what this would allow us to do is to advance towards full understanding regarding how the social determinants drive downstream biologic mechanisms of aging with regard to muscle health in order to intervene on health inequities. And this is really the focus of my K99R00 award. The title of my project is Examining the Role of the Neighborhood Environment on Muscle Function in Older Adults. And what's so exciting about this project is that it really is directly aligned with NIA's strategic goals, which seeks to integrate the social with the biologic, exploring the role of mitochondrial function as a way of ultimately intervening on health inequities. So just briefly here about the K99R00 mechanism. It's comprised of sort of two phases. The first is 2 years of postdoc training, which I'm in right now. And then I'll be segueing for the last 3 years of the award into the R00 phase, which is composed of 3 years of R-level funding. And really the main thrust of this mechanism is to transition early investigators to an R01 as quickly as possible. And so what I'm showing you here is the broad sort of conceptual framework of my K. And what I'm so excited about is that really what this project represents is sort of my first opportunity to really bridge these areas of understanding how the neighborhood environment influences downstream muscle function, and to what extent physical activity and mitochondrial bioenergetics may serve as key mechanisms in linking the neighborhood environment to muscle function. And so the K99 phase I'm working on right now, which is the aim one of this project, examining the relationship between neighborhoods and muscle function. And in addition, during this K99 phase, I'm currently in the middle of training in some really exciting areas around neighborhood measurement and geocoding, causal mediation analysis, skeletal muscle biology, and mitochondrial bioenergetics, which I'll be returning to shortly about that training. And so ultimately what this training is positioning me to do is that by the time I finish this phase, I will be equipped with the expertise in the social science of neighborhoods and mitochondrial function that will really allow me to sort of fully explore how social experiences get under the skin and influence numerous aging-related outcomes. For the R00 phase of this award, I'll be completing aims 2 and 3, which look at the relationship between neighborhoods and mitochondrial function, and then using causal mediation to test mechanisms of the neighborhood muscle function relationship. In order to answer the questions aims 2 and 3 for the R00 phase, I'll be using a really cool new data set called the Study of Muscle Mobility and Aging. It just got off the ground about 2 years ago and is a multi-center study at the University of Pittsburgh, Wake Forest, and then UCSF is leading the study and is overseeing all the data collection. SOMA is comprised of 1,000 older adults aged 70 years and older, and it's extremely well phenotyped with regard to physical functioning measures, muscle physiology, exercise, diet. Participants will be wearing wearables for an extended period of time, and it's just a very impressive study, and it has some very cool measurements. But perhaps I should say, when I started at UCSF, this project was just getting off the ground. So I've really been involved in sort of getting it off the ground, both in thinking about unique measures of the neighborhood that I've been able to add in. And so one thing that I want to sort of hone in